Becker, Zone Law, Chris Parkin, and the McDermott Institute. The other group we're relying on for this event is our judges. So thank you again. Right, I think our finalists are waiting. There will be an introduced as each of them. I don't know who is coming in. There's some noise.
Probably the Wellington wind hammering my house.
I don't think it is the Wellington wind hammering the, there seems to be a bit of a hiccup there. So, and I think I can hear somebody in the background. So we're going to move one of the first of our contestants up into the panel. So can we please introduce Shaolin?
Oh, hello.
Shaolin, welcome, and it's good to see you.
Hi there. Yeah, it's very nice to see your face again.
Shaolin is something's really coming through really weird.
Oh yeah.
I think Jack's got something that's not coming through clearly on the video. My apologies. Carry on, Shaolin. Shaolin is a year 13 prefect at Mount Auckland Grammar School. He's an advocate for the research and development of self-sustaining energy and the protection of New Zealand's fresh water. Apart from science, he loves to surround himself in the arts. He is also a musician and producer for the Meme Fashion Show in Portugal, a filmographer or cinematographer, or a hip-hop dancer. He holds a world championship title from 2019. Shaolin's STEM presentation topic is a sustainable future with biodigestion. Thanks, Shaolin. Over to you, Kim.
Thank you very much.
Yes, thank you, Shaolin. Interesting topic. Have you worked out what the carbon footprint of transporting all this waste to the digestion centres might be?
So I haven't thought of a specific number, or I haven't found a specific number for what the amount of, or how much CO2 could be produced in terms of how it's transported. However, one of the really nice things with biogas coming from biodigestion is that a lot of it can be used as a alternative for normal gas in vehicles. So I know in Berlin, with the current biodigestion methods that they're using, they've had them set up and they've got their brown bin system, but all their vehicle transportation, or all their vehicles that they use to collect the waste actually run on their biogas, so they don't actually add any extra CO2 pollution.
And in Berlin, it's collected from domestic and commercial premises.
Yes, they do have both on that end.
The bacteria, the anaerobic bacteria that you require for your digestion, they're fairly well known, are they?
Mm-hmm. Well, I haven't done too much research into the specifics of what bacteria is used in where, but I think it's a very fundamental concept of biodigestion, and I don't think it's something that will change that much over the years. So I think as biodigestion comes in, I think bacteria is going to be the last thing that's to change at all, because it's a very fundamental sort of chemical process. So yes, I would say relatively that'll stay put.
The resulting sludge you're suggesting would be used as fertilizer.
Yes.
Would that be acceptable to Maori?
Would that be acceptable to Maori? I have not looked into the social implications of using digestate and whether or not that may affect Maori beliefs. That's not something that I have researched myself particularly. However, the use of digestate, I think, is very variable. So I think that it may be possible that there are certain uses of digestate that the Māori community might be against, but I think that's also the beauty of the digestate that comes out of biodigesters, is that there's lots of different ways that it could be used. So we could use it as bedding for livestock. We could take the liquid out of it and then use that as a high concentrated fertilizer, or we could actually just send it back into the composting system and then let them work with that. So I think if there is something that might be against the use of digestates, I think there's a lot of options out there that might be acceptable.
Can you tell me a bit more about the gas? How will it be captured? What will happen to it, the biogas?
So the biogas in itself is mainly made out of CO2 and methane. So there's a lot of ways that there's a lot of things that you can use the biogas for. So the most common one is that it's used for co-energy. So it's just used to burn, it's burnt, and then it's used for more energy. So the Mangere wastewater treatment plants, they collect effluents and fecal matter from New Zealand, and then they will create biogas from that, and then they use that to co-generate the plants. So that's one option that you can use the gas to use. There are also some other ways you can use it. You can separate the CO2 and the methane and use that for different options. So I know in Reparoa, we have our big biodigester that's currently being used in the works. And what they're planning to use the gas for is they're going to separate the CO2 and the methane, and then they're going to pump the CO2 into some glass houses, which they'll have some tomato plants growing. And the CO2 that's being pumped into that will greatly encourage the production of those plants. And then from that, you can get the tomatoes and you'll have more organic waste to put back into the biodigester. So it's a nice little positive feedback loop there.
Have you done any maths on how it would affect the greenhouse gas emissions in New Zealand? Were your proposal to go ahead, Auckland concentrating?
So I haven't done a huge amount of calculations on how much greenhouse gas emissions it would add. I have done some calculations on how much gas we would be creating to be used. So I've done a look into a couple places. So I first looked off into France because they have some open resources available. So just for a bit of reference, the digesters they have over there, they can use, with 1 cubic meter of gas that they produce, you can make around about 6 kilowatts of heat energy. And with their digesters, you can produce anywhere between 20 to 800 cubic meters of gas per ton of organic matter. So let's say if we gave it 230,000 tons of food waste, you'd be getting anywhere between 4.6 and 184 million cubic meters of gas that could be used. So that's something that's happening over in France. Within New Zealand, it's been a little bit difficult to find out the information in terms of how much gas could be produced from food waste and organic waste, because it seems to be quite commercially sensitive. So there's not a lot of information we have within that area. I do have some more information, however, on Mangere, because they deal with effluents rather than the food waste. So for Mangere, they deal with sewage. And on average, a person makes about 130 grams of fecal matter per person per day. From that, you've got about 140 grams of chemical oxygen demand per person per day. So in Mangere, with every 140 grams of COD, you could make about 30 liters of gas a day. So a lot of potential for the amount of gas that could be produced. But unfortunately, I have not taken a look into how much that would be adding to overall pollutant of gas.
Because just to be clear, the gases you're producing are greenhouse gases, right?
Yes, CO2 and methane.
So it's renewable energy, but it's not climate friendly.
Well, it's a lot more climate friendly than a lot of alternative options that we have out there. So obviously a big competitor that we would have would be composting, because that's something that we already deal with quite a lot. And main differences between composting in terms of how they deal with their gas and whatnot is that bacteria in biodigestion is anaerobically used, and in composting, it's aerobically used. So that means it's basically open to the general atmosphere. So when you get all your organic matter and you put it into a compost, there's usually these long sort of tubes which they'll put things into, and then they'll have these little holes to allow oxygen to come through. And that's really good. However, because composting still does produce bits of CO2 and methane, and they also produce a little bit of nitrous oxide, and that's due to some aerobic that's due to some aerobic digestion that's happening in little pockets of the composting where there's not enough oxygen to get through. So that's how that gas is created, and then that'll eventually seep out of the holes that are within the composting, and that just goes into the open air. So I think something that is an advantage with biodigestion is that we use that gas. We take the methane and we take the CO2 and we use it to create biogas and other things. So yes, it does create more. It does create more greenhouse gases, but the big thing is that we capture it and we use as much of it as we can compared to just letting it go out into the open.
Thank you. Thanks very much.
Thank you very much.
Joy, you're muted.
Our next finalist is Kunley. Kunley is an avid participant in a wide range of activities, from chamber music to tramping to debating. He has always been particularly interested in how scientists could use their knowledge to benefit our natural environment. Kunley has a breadth of leadership experience within the school and the wider community. He's a Year 13 student and Deputy Head Boy at Kristen School in Auckland. Kunley is looking to study computer science and engineering at university, combining his interest in the sciences and technology and adding his enjoyment of public speaking to design and to deliver practical and efficient solutions to problems of the 21st century. Kunley's STEM presentation subject is Something Sweet, the Sugar Biobattery. Over to you. Thanks, Kim.
Hi, Kunley. Are you there?
Yes, I'm here. Hi, nice to meet you.
Jolly good. The biobattery seems to need a lot of corn, right? It needs corn to produce.
Yes.
Won't that put pressure on the agricultural land and the food supply?
So there are a number of different ways that we can produce maltodextrin. So one of the ways I talked about in my presentation was using cornstarch. That's one pathway that you can use to get to maltodextrin, which is the fuel that we use to power the fuel cell. There are other ways, so we can come up with pathways. Another way I talked about was using cellulose, which is something that can also be used to generate maltodextrin. So yes, while it's true that we will need to increase our corn production, because of the fact that we're generating electricity through corn, that means that we do also have the opportunity to reduce the use of coal, for example. And that obviously has a net benefit for our environment because of the fact that we're reducing the pressure on on mining or drilling, and rather we're transferring it to something that New Zealand is relatively efficient at and we're quite good at, which is agriculture.
Is this battery rechargeable?
Yes, so this technology is really unique, and the best analogy is that it's similar to an ink cartridge, which is that once it runs out of ink or once it runs out of power, you just refill it with maltodextrin and it's able to continue operating and continue generating electricity in that way.
I mean, enzymes are complicated things. Have we got the facility to use them to perfect, if you like, because they've got, as you point out, a very short life?
That's a really important point, I think, and this technology is still relatively in its infancy. So that's one of the things that we do need to look at, which is how can we ensure that the battery can sustain itself, have a good life, as in the enzymes don't die off or denature. And so that is something that scientists should be looking at and are looking at this stage, which is how do we extend the life of these enzymes so that the battery can actually be sustainable and applied commercially.
Any ideas about that? How could you extend the life of the enzyme?
So there are a number of ways. Thinking about how to ensure the chemical environment within the battery is maintained. Thinking about ways perhaps to replace the enzymes if they do denature and they are no longer efficient. Those are some ways that the enzymes we could ensure that the enzymes do stay efficient. But yeah, that's definitely a challenge that scientists are looking at right now in this technology.
What does the battery look like? I mean, what's it encased in?
So the battery itself is actually quite, I don't know if you've seen pictures of like hydrogen fuel cells, like those, like they're a bit clunky right now, obviously, because it's quite a new technology, but it's not doesn't quite look like a regular battery with a metal casing, but it's like a device that, like a larger device where it has a fuel cell. It's a bit like a mini generator, if you like. That's what it looks like in its current stage. And
the environmental impacts of it, obviously less toxicity, but also, as again, I return to the fact that you're going to have to use a lot of land. Whatever you use for the fuel, you're going to have to devote a lot of land to growing it, unless you use waste products.
Yeah, so I guess that like land will always need to be used to generate electricity. I don't think we can get away with that. The fact that we will never, like, we won't be able to generate electricity without using land, but the benefit of using a source, which is like when you grow corn and you use the land to grow corn, that corn can be used in a number of different ways. It doesn't have to go towards electricity or energy production. So it means that we won't necessarily have as much waste as if we use that land to have maybe solar panels, which might become outdated, or they might, like, you will have to eventually throw them away when they become inefficient. If we're using that land rather to grow corn and to use corn to generate our electricity, I think that gives us a lot more options for that land, and it's also probably a better use of that land anyway.
What about timber byproducts? Are they a candidate for this?
Sorry, timber?
Timber byproducts.
Yeah, so one of the benefits of using EFC technology, enzyme fuel cell technology, is that as long as you can come up with the correct enzymatic pathway to get to your product, it means that you can use that as a source of fuel. So one of the challenges of the industry is coming up with different ways to make different products in efficient ways. So if it is the case that New Zealand does hop onto this technology and we have a lot of waste timber, for example, then something our scientists could be looking at is how can we use that waste product, use a similar enzymatic pathway and reach the product so that we can generate electricity in that way.
And the byproduct of the battery is water, right?
Yes.
So what happens to the water?
So it's released as steam, like vapor.
All right. So that's entirely compatible with using the battery in appliances?
Yes. So that is something that can be looked at. So it's similar to hydrogen fuel cells in that way, I guess. But yeah, the fuel cell does need to breathe, so it does need to have oxygen being fed into it. I guess if it is the case that that battery becomes efficient enough to use in portable electronics, I'm sure that we could come up with a way to have a vent or something for that, for your calculator or something, to have that battery being used.
Can you just explain simply to me what exactly the enzymes are doing when they convert the energy into maltodextrin into electricity?
Yeah, so different molecules store energy in ways. So they store energy within those molecules. Enzymes basically break apart that molecule, and by doing that, they're able to extract energy from the molecule. And through that process, that energy can be collected through a device that the scientists have created, and that generates an electrical current, and that generates electricity.
How widespread do you think the use of this could be?
Well, it is still very, very new technology. There's a lot of different alternatives to this. This is just one of the ones that I looked at and talked about. But I think if it is successful, this could be something that replaces all of our batteries for our personal electronics in the next decade.
Cars.
Yeah, so cars might be a bit far off, but definitely in the future, we could have cars that run off this technology, similar to hydrogen fuel cells. We could have cars running off enzymes. Yeah.
Thank you. Thank you very much, Kunley.
Thank you, Kunley. Okay, our next finalist is Rohit. Rohit Gupta is an 11-year student at Scots College in Wellington. He has a keen interest in the sciences and supports Eureka's aims to demonstrate how the application of science and technology can benefit New Zealand. He is also an active debater and cricketer. So Rohit's STEM presentation topic is nitrate leaching, a soil-based solution. Thanks, Rohit. Thanks, Ken.
Hi, everyone.
Hi, Rohit. Thank you for that. You're inhibiting the activity of nitrifying bacteria by nitrification inhibitors. What are the inhibitors?
Essentially, the inhibitors are these chemical compounds that need to be understood. They're simply compounds which you apply to the soil in the form through a spray or through a distributor. The compounds have been designed to target or to apply their function to these ammonium oxidizing organisms, or these nitrifying bacteria, as you said. These chemical compounds have been tested and they have no environmental impact on farming microbes or probiotics in the soil. So they don't have a negative impact on the soil. And their purpose is purely to target these nitrifying bacteria, or these ammonium oxidizing organisms.
So do we have the inhibitors already? Do we have to make them? Is it a chemical process? How do you get them?
Yes, so basically we have around 8 presently potential inhibitors. These are general compounds. They could be applied in many circumstances. However, studies have shown that if a country or an initiative were to put enough effort in, they could tailor these chemical compounds to suit a particular environment, because naturally all sorts of countries have different farming situations and different ecosystems. And in New Zealand, we have our own diverse and unique ecosystem, especially when it comes to farming. So if we were to put effort into tailoring our own inhibitor based on these sort of general compounds, we could increase productivity and actually decrease costs and increase the effectivity of these compounds, which would be beneficial for everyone. So yeah.
You're suggesting that our water quality is pretty high already.
I'm suggesting that our water quality is internationally in a decent position. However, if we're flouting this image of being 100% pure, when behind the curtains, 2/3 of our rivers are too polluted to swim in, 70% of our fish are threatened species, I don't think we can make those claims. And I wish I could make those claims to be proud of our nation's our river situation, but unfortunately, that isn't the case. And I believe that we are leading the world in many ways in sustainable farmings, but that doesn't mean we can get complacent, especially with the present state of our lakes and rivers, which I believe could be better. And I think that there's no reason for us not to strive for even better standards and even better river qualities, which I believe should be the ultimate goal for a country which proclaims itself to be green, clean, and 100% Middle Earth, because I think 33% pure doesn't roll off the tongue just as well.
Inhibitors have acquired a bad reputation in the past. People are suspicious of them.
Yes,
How would you turn that around?
So essentially, yeah. So in 2013, there were fears that DCD residue, which was one of those general inhibitor compounds, was appearing in milk. And because of that, because of the fact it was appearing in the media so strongly, even though, of course, farming uses all sorts of compounds and it's not completely pure anymore, because of that media attention, DCD was completely just suspended across the country and it just plummeted. And it's never really regained any footing, even though in November of 2013, so later that year, the Ministry of Agriculture and Primary Industries performed rigorous study on the DCD inhibitors to actually discuss, because they had already done reports on DCD and quoted beneficial effects, and they actually encouraged the use of them. So they did another study looking at milk, and they actually found that the presence of DCD was so minimal as a residue that it actually posed no food safety risk whatsoever. I think if we market these inhibitors from the lens of their environmental impact, which is massive. The trials conducted saw 80% decrease in nitrous oxide emissions and 50% decrease in nitrate leaching, as well as increases in herbage productivity. If we market those beneficial impacts, I think we can definitely turn around their image, especially because the government has actually stood behind these inhibitors, even though they haven't actually returned to New Zealand as of yet.
How many soil-based nitrifying bacteria are there, species-wise?
There are quite a few. I think the most well-known is Nitrosomonas. It's most present in the soil. It's a group of bacteria which especially target ammonium, but these are microbes, and ammonium oxidizing organism is quite a general term, but most of these inhibitors are designed to target Nitrosomonas and similar microbes and similar bacteria, because those are the most well-known and they're the most effective when it comes to transforming ammonium into nitrates and then nitrate into nitrous oxide as a greenhouse gas or nitrate as a pollutant in our waterways.
So how specific would your inhibitors be? Because you're messing with soil, right? And there's a lot of stuff in soil that's very, very useful.
Yeah, naturally. Yeah, definitely.
Can you target these?
Yeah, so obviously it would be very difficult to get down to the individual mutation of a certain bacteria, but we can target species. And these inhibitors were designed not to touch farming microbes and not to touch probiotics in the soil, because naturally a soil ecosystem is very complex and you can't just go around wiping out microorganisms in the soil, because that would just be devastating. But we can target these species like Nitrosomonas, as I said, which would definitely benefit the soil in terms of how effectively it could retain ammonium, which is the ultimate goal here, I suppose.
Resistance would be an issue that you would need to think about, would it not?
Yes, of course. Yeah, the bacteria could potentially mutate and that resistance would have to be present. But as I said, these inhibitors are really flexible, and if enough research is done, you can actually tailor them to specific environments and, as I said, specific species, which I believe is definitely a hurdle that we can overcome if we dedicate enough resources towards it.
And what would farmers spray them on? I mean, how are they applied?
Yes. So essentially in the Bay of Plenty trials, which is what I based most of my presentation of, they were used in a machine. Essentially the machine had sprays and it also had sensors, which was actually a crucial aspect. Urine patches only cover about 4% of the average farming paddock. So it would be completely wasteful for us to go around entire farms spraying inhibitors everywhere, when inhibitors are only really needed on 4% of a farm's area. Of course, that 4% is incredibly potent, but it is only 4%. So the sensors and the machine that we used in the Bay of Plenty trials basically covered the farm, found these urine patches, and applied the inhibitors where was needed. And that's excellent because it's efficient and it doesn't require us to, because imagine how much labor it would be on farms of massive scale to apply these inhibitors just universally. But with this technology and these sensors, which have been developed very recently, you can actually detect urine patches, which is where these nitrates are supersaturated. And there you can apply the inhibitors, which is just great because it completely streamlines the system, makes everything more efficient. And yeah.
Thank you. Thanks, Rohit.
Thank you.
Thanks very much, Rohit. Well done.
Thank you.
So our next finalist is Ethan McCormick. Ethan is a year 13 student from Howick College in Auckland. He has a keen interest in New Zealand environment and is also involved with area restoration and pest-free initiatives in his local community. He is active in debating, student leadership, and running groups within his school. He plans to attend Auckland University to study history, law, or science, to develop his interest in managing the environment through leadership, while also pursuing his interests in writing and communication. Ethan's STEM presentation offers a diagnostics for managing cowrie dieback, which is a topic he has investigated for some time. He is actively seeking solutions to protect New Zealand's native heritage. Ethan's STEM presentation topic is catching the cowrie killer. Thanks, Ethan. Thanks, Kim.
Hi, Ethan.
Hey, Kim.
I just got back from the cowrie forest up north, actually, so I was interested in your presentation. I can't see your picture yet.
Oh.
Don't know whether to wait for it or just carry right on. Let me ask you, this is...
he is there.
This offers an opportunity for citizen science, essentially, with your lateral flow devices.
Yeah.
But you actually don't want people tromping around the cowrie, albeit with the best of intentions, do you?
Oh yeah, totally. I mean, I think there will be a degree of spread if we've got people going out there and using these tests widely. But I think that if we can have, say, community groups who are following regulations and following guidelines, we can have, say, regional authorities such as local councils, they can authorize such community groups to go out there and follow the guidelines, just so long as people are going in between trees that they test. So just so long as they're spraying their footwear, scrubbing their footwear, it will reduce the spread, I think, to a degree. And I think that it is a case of pros and cons. We've got to look at the big picture here. I think that if we weren't doing anything, or we weren't testing, that the spread would be a lot worse compared to if we're able to pinpoint it to individual trees. And I think that if we're able to do that and able to pinpoint it to individual trees, then it will be a far greater impact, and we'll be able to save large swathes of Coady forest. So yes, there will be minimal losses perhaps, but I think that in the long run, it's beneficial.
As a matter of interest, you make the point that it's important to find out whether Kauri dieback has been introduced or here all along. Why does that matter?
Well, I think it's important to know how specifically the disease behaves. So a lot of other Phytophthora species, such as Phytophthora infestans, which cause the Irish potato famine, that can also be spread by the air. It can be spread as an airborne pathogen. Coady dieback, they can conclude with a great degree of certainty that it's not, and it's just spread via the soil. But having an idea of how invasive the species is and how quickly it can spread will give us a better idea of how to contain it.
If the lateral flow device is available already, why isn't it applied to Kauri? It's used for other things, right?
Yeah. So the important thing here is that Coady dieback is that the generic test that can be bought online, really easy to get. But the problem is that it's not specific enough for what we're looking for. So there was a study done by Auckland Council in 2018, and they found that 7 out of 13 of the sites that they sampled that were positive for Coady dieback also had Phytophthora cinnamomi, which was one of Coady dieback's close cousins. So using that generic test, we'd also be picking up Phytophthora cinnamomi, and we'd be getting really an accurate result. So that's the awesome thing about having a really specific device, that we can conclude that if we get a positive result, it is a positive result and it's not a false negative.
And mice produce Kauri dieback antibodies.
Yeah, so the process of producing antibodies is a general process, and it can be applied to all sorts of things. So whether that be human-specific diseases, plant diseases, animal diseases, it is a generic process. And so essentially what you'd be doing is you'd be introducing the pathogen to the mouse, and the mouse's immune system is known to be naive. So it will generate antibodies to any pathogen that is introduced to it. And it has been done before plenty of times on other Phytophthora species, such as I mentioned in my presentation, Professor Hardham from Australia, who's done it on Phytophthora cinnamomi, which is a really good case study for us to look at because it is so closely related to Coady dieback.
All right, so you've got, if this worked, a quick diagnosis of Kauri dieback disease caused by Phytophthora.
Yeah.
But you still don't know what to do about it. And the only treatment, it seems, is phosphite at the moment.
Yeah.
So will we be any better off?
Well, the important thing is to stop the spread, and that's the main weapon that we've got right now. So it's Rahui and track closures. Those are the two main things that we can do to stop Coady dieback. And it may well be that in 10, 20 years, or maybe tomorrow, that we've got a cure, but until then we don't. And so up until that moment, it's important that we're stopping the spread as much as we can. And I think the cool thing about this device is that it can pinpoint the spread to almost to individual trees if it was, say, if you've got a high enough level of community testing. And if you're able to do that, you can tell people not to walk in specific areas and spread it. And you can get rid of other minor vectors as well, such as feral pigs and perhaps possums. So I think that, so hopefully we will have a cure, but until then, I think we've just got to keep doing what we're doing at the moment.
What is it, Phytophthora? What is it?
It's a bit of a weird species. It's kind of neither, it's classed as an algae, but it displays attributes of fungi and bacteria at the same time. So as we've seen with the Irish potato famine back in the 1840s, is that was spread via airborne spores, and Coady dieback is spread by waterborne spores. So it actually releases spores, yet it's an algae, and it kind of and it also multiplies as well, similar to bacteria. So it's an odd disease, but it has been around for a long time, and I think it's, and as humans, we've obviously learned to live with it to a degree.
Thank you, Ethan.
Thank you.
Thanks, Ethan. Okay, so our next finalist is Cynthia. Cynthia Zhang is a Year 12 student at St Mary's College in Auckland. She's an avid learner who likes to challenge herself so that she can always do her best, being top of her cohort since 2016 and being selected for a number of STEM awards and scholarships. She loves to get involved in any opportunity in her community and is an active member of initiatives like organising committees for key events, youth councils and orchestras. In her spare time, you'll find her either reading books, learning about news discoveries, or exploring the outdoors with her camera. Cynthia's STEM presentation is Saving Our Native Birds. Thank you, Cynthia. Thank you, Kim.
Thank you.
Hi, nice
Kim.
of you to join us on this Zoom. What specific damage is done to native birds by possums? Do you know?
Well, possums harm native birds in a variety of ways. Not only do they eat their eggs, they also eat their like young possums as well, and obviously they also harm native birds in a variety of ways, such as killing them, and also they carry diseases which can obviously not only harm native birds but also harm a variety of crops such as like meat and dairy. So they do a variety of damage.
I mean, you focus on possums, but of course a great deal of damage is done by other introduced species as well.
Yeah, definitely. Introduced species harm a variety of ways. I just focus on possums for easier understanding.
All right. The poison and the traps don't do it then, so you want genetic engineering.
Yes, essentially.
And New Zealand has been very averse to genetic engineering. That's a major problem.
Yes, that's true.
How
That's
would you
obviously
overcome?
Well, obviously the major issue at the moment with gene editing, a lot of people are opposed to it is because it's quite uncertain. It's a new technology, and obviously with every new technology, people are genuinely going to be opposing it. But I feel like with a lot more, if we do a lot more research and a lot more development towards gene editing so that people are better educated about the impacts of it, I feel like people will be more acceptable about this whole gene editing issue. And also I think it's really important at this moment to be very open and minded about new solutions, because this solution with gene editing could definitely impact New Zealand's environment really beneficially if it's used successfully. So I feel like with just a lot more research and a lot more education, everyone will generally become more aware and they'll accept it more than what's happening right now.
How would we do more research that would enlighten us as to the scale of the impact on the environment of this kind of gene editing?
Well, we could do more research through a variety of ways. So basically, we could get more people on board of this whole situation and implement this process. We could invite different people to come over to New Zealand to help with the whole process. Most importantly, though, at this point, it's just important for everyone to get into this new situation together and obviously work together to help. That's probably the best approach for the situation.
What you're advocating is propaganda, not research.
Not exactly. I feel like with having more people coming over, they are able to collaborate, and that's basically the essence of science. You have more people sharing ideas and sharing different ways to research things. And then once people come together and share these ideas, you're advocating for better research quality than what's happening right now.
You've suggested that the cost of this, using this particular SRY gene, is $9 billion over 50 years.
Yes, that's correct.
Yeah.
Oh, I personally didn't work it out. I read a research paper written by James Russell and a couple of his colleagues, and he did all the math together. And so basically he wrote out a paper about it. He's a young ecologist from New Zealand, and that's where I took this idea and sum from.
If we were to use this technology, how many possums would you need to amend in order to have any impact on the population?
Well, Firstly, before we can actually think about amending possums for like a large impact for the whole of New Zealand, I would say typically we need to start going very small and focusing on the small island. So I would suggest amending maybe around about a really small quantity, like around 100 or 10 to 15 or so. I'm not generally sure about the actual answer to that, but like just amending a small amount and focusing on a small area to actually observe the impact is important before actually going full scale.
And do you think that this would be acceptable to Maori?
Well, the one important thing for the Māori is kiakitanga, which is basically guardianship. And so they have this really important role of protecting the environment. And considering introduced predators do harm the environment, I think this will be having procedures to decrease this amount of introduced predators and have a better impact on the environment is something they really value. So I think this would be acceptable to the Māori as we are actually helping the environment as opposed to destroying it.
And what's your understanding of how Mātauranga could fit with this?
Well, basically we could have Māori. So firstly, we need to make sure that we actively involve them in discussions and like actually communicate to them as opposed to just going with this and not thinking about them, obviously. So we need to communicate with them. And also we could have certain Māori people as key like experts to guide scientists and make sure they take a good approach instead of like harming New Zealand's environment, as obviously that's not what we want. And so by ensuring that we involve Māori people and Māori scientists, we can achieve Vision Mātauranga.
So you're talking about selling the concept to Māori.
I'm not talking about selling the concept. I'm more of like you're involving them in discussions, and you're not trying to like persuade them. You want them to actually agree with this and see the benefits it could bring to New Zealand, as opposed to going, "Hey, we want to do this. Can you agree?"
Can you see any downsides to it?
Well, obviously with every solution, there are downsides, and possibly, obviously, we need a lot more research, what comes with the cost and economic consequences, and there also the amount of communication required and probably the time for this overall process. But considering the long-term impact on the benefits to New Zealand, which is immense, we've got impacting the environment and the economy in like a lot of beneficial ways. I feel like the benefits override the consequences. I feel like this definitely should be considered.
And you can't see any risks of using this kind of genetic modification?
Well, obviously with gene editing, as it's really uncertain, there are risks and things to consider. So I feel like slowing down and taking important measures along every step and thinking of different things to implement to make sure that we don't actually use gene editing to harm anyone is really important.
Appreciate your time. Thank you.
Thank you.
Thank you, Cynthia. Our last contestant or last finalist in the secondary schools is Cullen Tran. Cullen is a Year 12 student at Scots College with a passion for science and chemistry in particular. Beyond the classroom, he's also in the school choir, a member of the Environment Council, and a voluntary tutor to younger students. He is a strong advocate for empowering youth, campaigning in his free time and being a member of the debating team. Eureka has been an opportunity to combine his love of public speaking and science. Cullen's STEM presentation is Healing with Hydrogels. Thank you, Cullen. Thank you, Kim.
Thank you, Joy.
Kia ora, Cullen.
Hi, Kim.
What are you doing? You're jamming a piece of hydrogel into the brain, essentially.
Well, basically there's two purposes. So if you're focusing on emergency treatment, then you're using hydrogel patches. So they can really be applied anywhere on the body, but because the brain's often irritated because of the stroke, you'd apply it to the arm, and then the growth factors such as VEGF and IGF travel through the bloodstream to the brain to grow new blood vessels. But I did also cover the other purpose, which is as a rehabilitation tool. So if you wanted to use them to grow new blood tissue, then yes, you do an injection of hydrogels as a scaffold in the brain. It just depends.
And to what extent are they being used in New Zealand now?
In New Zealand, the hydrogel industry really hasn't taken off, and that's largely because of the high cost to make them and the limited commercial investment right now for medical properties, because the status quo means they're prevented from going into an industry which is economically viable. But in New Zealand, we're currently using them for more sort of trivial uses, such as like external wound healing and dressings. So yeah, we haven't gotten there yet. So New Zealand still needs to take that big leap.
Why do you think that they're not being used in this way?
Well, firstly, because of the high cost, but also because quite a lot of the research has been done overseas. And I don't think New Zealand's really recognized the extent to which stroke is impacting us. I think a survey studied last year of about 400 people in New Zealand, only about 1.5% identified stroke as like a major disease, in comparison to like 37% for heart disease and 33% for cancer. So I just think we've got a lack of awareness about how much stroke is really impacting us. And that's why we haven't really turned to hydrogels as a solution for them.
So let me get it clear. You've infused your hydrogels with growth factors.
Yeah.
And if I'd suffered a hemorrhagic stroke,
yeah,
what do you do? What do you do then?
Okay, so I covered mostly the ischemic strokes because ischemic and embolic strokes make up about 85%. So that's
okay. I've just had an ischemic stroke.
Okay, so for an ischemic stroke, because of the way that the patches are designed, if you're talking about you've just had a stroke, then what that means is because you don't need a specialist to apply it. So in terms of the other treatments which exist currently, which is TPA or emergency surgery, that has to be administered at the hospital. So these patches can be taken like in the ambulance and applied on site, on delivery on the way to the hospital, so that blood vessels start forming. It only takes about 24 minutes for there to be significant progress, and then about an hour and a half for new blood vessels to have completely.
And this is as the result of the ingredients from your hydrogels going to the required area through the skin.
Yeah. So the hydrogel.
So why don't you just inject it? I mean, why hydrogel?
Okay. So the whole reason you use hydrogels is because, we could already use VEGF, but the problem is that it has such a short half-life. So if you just inject it normally into the bloodstream, then it's going to degrade within 30 minutes, like really rapidly. And the point of the hydrogels is that they sit under the skin and they release a really controlled flow of the growth factors, which means that they can cross the blood-brain barrier effectively, and you're getting the results you want without causing other side effects, which can be produced if you did such a large dose at one time. So they're more like a mechanism for the growth factor.
How do you get it under the skin?
So you use the microneedles. So why we wouldn't use a normal needle is because that already affects the blood vessels are there. So you're damaging what you already have. But with microneedles, it allows the hydrogel to just cross the very outermost layer. So they sit under the skin, and then they can release the growth factors without harming anything else already in the body.
And is this used in this way anywhere at the moment?
It's been done in a lot of trials, but because I don't think it's been recognized as something that has been implemented as a proper official treatment, it's not been implemented that well.
And it involves stem cells.
Yeah, so if you're using stem cells, that's more of a rehabilitation process. So after a stroke's occurred, yeah. And then if you want to grow back blood tissue, brain tissue.
Do you think that applying the hydrogel very quickly would halt the stroke or improve rehabilitation down the line?
It's both. So that's why I kind of covered both of the uses in my presentation. So in terms of emergency treatment, it's really good in transit, because it's perhaps not really a tool that can be used solely on its own in some cases. Sometimes the clot in the brain is too big, and it needs to be removed using traditional treatment, which is like a stent retriever, which was where they stick the sort of metal straw up your femoral artery, and then they remove the clot. But in terms of being able to provide that sort of care for patients, because the main challenge facing stroke victims is the idea of getting to the hospitals and appropriate care on time. About only 33% of patients arrive under that 3-hour window that is needed for effective results. And then in saying that, even after the strokes occurred, and even if there's been like lots of damage done, they can be used and infused with these stem cells to grow back the brain tissue, which other treatments don't have the capability of doing.
Why have you got such an interest in strokes?
Personally, I've had family members in my family affected by stroke. I have a lot of family friends. I've seen a stroke happen to someone, so obviously it had a very personal impact to me. And obviously when I researched a bit more about it, I found out how detrimental it was to New Zealand. I really didn't realize it costed us about 3 billion each year. So yeah. Obviously hydrogels have different purposes. You can use them for diabetics with insulin and other drug delivery, but I focused on strokes because I thought that would be the most relevant issue.
Thank you. Thank you, Cullen.
Thank you.
Thank you, Cullen. Now we move to the tertiary undergraduates. And our first finalist is Karen Banker. Karen's a fourth-year medical student at Auckland City Hospital. And prior to medical school, he completed a degree in neuroscience at the University of Otago. He's particularly interested in surgery as there's an ability to make large differences in people's lives in a short amount of time, which he finds hugely satisfying. In his free time, he plays cricket or hikes the stunning New Zealand landscape with his camera. Karen's STEM presentation is The Future of Medicine, Organs on a Chip. Thanks, Karen. Thanks, Kim.
Kia ora, Karen.
Kia ora, Kim.
Where do the cells come from that you make the chip out of?
Yeah, so the cells are human cells, and we can obtain them from doing simple things like taking them from skin, doing little biopsies, and basically it depends on the organ that we're trying to mimic. So if we're trying to mimic the lungs, then obviously we would need to do some sort of biopsies, getting the epithelial cells or lung cells from inside, and then putting them on the chip.
Who's going to volunteer for that one?
Who is going to volunteer doing it, or
donating bits of their lung for your chip?
Yeah, so obviously that would need some recruiting, and hopefully we would reach a stage where we can replicate these cells inside the lab so we don't have to continuously take samples from humans. Yeah, so ideally we will have like a bank of human cells that we can continuously use to create these organ chips.
That's opens up an ethical can of worms, does it not?
Yes, it does.
Have you addressed those?
So what sort of ethical principles were you talking about?
Well, I imagine that Maori might not be happy growing human cells in a lab, for example.
Yeah, so we would definitely have to take into consideration the Maori worldview, see what they would want in this sort of experiment. We have to take into consideration the benefits, the negatives. But yeah, there are some ethical issues. I'm not saying that we have currently reached a stage where we're continuously creating cells inside the lab. It's just an option.
How soon, I mean, I take your point that we're facing a COVID pandemic. If we could develop a vaccine quickly by means of an organ chip, then obviously that would help quell it. But you would still need to have clinical trials on people. You would still need to test it on animals. Would you not?
I think I agree with the humans, but to a certain degree. I definitely don't agree with the animals. I think animal testing we're reaching a stage where animal testing is being significantly reduced. And I think in the future it will go out. Whereas in this idea, I think humans will still be involved in clinical trials, especially for testing out medications and agents, but I don't think it will be to the degree that we have currently, because all the processes around research and development revolves around clinical trials on humans. Whereas if we had this technology, trialing on humans wouldn't be that significant. It'll be more of a surveillance sort of issue rather than seeing what the drug is doing to the human.
You don't think that the chances are that whatever you've done to your chip would behave very differently in the wider human population?
No, that's exactly what we're trying to mimic in this organs on a chip, is that we're trying to mimic exactly what happens inside the human body. So it's not just some cell that we're putting on a chip. It's not some static sort of experiment. This is a dynamic process. So this is the cells, all the layers, all the nutrients, everything that happens inside the human body. We're mimicking that to almost exactly the same thing on a chip. But you're right, once we trial it on the chip, then we will need to see what effects it has on a human, but we won't need the same amount of research and people needed.
I don't know, your chip seems remarkably simple. A human is incredibly complex, and a human in the environment is furthermore incredibly complex. I'm not sure, I remain to be convinced that your organ on a chip is going to cut the time and the cost involved in developing, for example, a vaccine.
Yeah, I think it's not really an organ on a chip. I just use that title, and I use that specific lung on a chip example in my talk to sort of illustrate the point. But I think a lot of experiments and research now is looking at integrating organs together on a chip. And so they're trying to come up with a human body on a chip. So it's taking...
body on a chip. Yeah, got you. All right.
Yeah, so it's addressing your point. You're right. The human body is just not one organ. It's made up of multiple different things that all interact together. And that's what now science and research groups are trying to do, integrating everything together and seeing how everything works together when you introduce things like medicines.
So we're looking at creating an artificial human being to test medicine on it.
It's not the same as like a clone or anything. It won't have the ability to think. So we don't have a brain. I think developing a brain on a chip would be extremely complex. And then that will go into even more ethical issues. Currently, it's just looking at the simple organs like kidneys, lungs, and liver, and some of the urinary organs as well. So yeah, I don't think we have a complete human body at this stage, and I don't think we will. But it is taking into consideration most of the organs and how they react.
And so the development of a vaccine this way would, I imagine, be very complicated.
Sorry, what was the question?
The development of a vaccine by means of this chip would be complicated, would it not?
Yeah, I mean, any sort of medication, developing it is complicated. But this idea is just so that instead of all the processes that we have currently using animals, recruiting people and going through all the lab testing, this will significantly reduce it. So by the time that we reach trials on humans, we've already got all the important data. We know exactly what will happen inside the body. Now it's just about testing it out. So we've taken away all the other steps that you would normally take on animals, etc. And we've sort of, fast-paced the testing process and creating a vaccine. But it will still be complicated.
What could an organ on a chip, for want of a better expression, tell you about?
Sorry, I couldn't hear that.
What would your organ on a chip tell you about side effects, for example?
Yeah, so that's another limitation. The organs on a chip wouldn't be able to tell you, oh, I'm feeling pain, or I've got a headache, or anything like that. But it will tell us a lot of the mechanisms of the drug. So, for example, absorption, toxicity, and things like that. And it will tell us the physiological and pathophysiological mechanisms. And from that, logically, we can sort of deduce what sort of impacts and symptoms this would produce if it was inside a human being.
Thank you. Thanks very much, Karen.
Thank you.
Thanks, Karen. Our next finalist is Robbie Morris. Robbie's a second-year student at the University of Waikato, and he's working towards a co-joint Bachelor of Business and a Bachelor of Science degree. He has the goal of integrating these areas to solve global issues. He's also a proud football referee, spending a significant portion of his time either training or refereeing. Robbie's STEM presentation is, What Can Seaweed Aquaculture Achieve? Thanks, Robbie. Thanks, Kim.
Thanks, Joy.
Hi, Robbie. Why did you choose a Colonia radiata as a target species for this?
Yeah, hi Kim. There's a few reasons behind it. The most important reason is, well, there's a few important reasons, but importantly, it's already got a market established in New Zealand. So there's a company, Agrisea, which I brought up in my presentation, who already use a Colonia radiata as biostimulants and as soil additive products. So they've already indicated that they would purchase a Colonia radiata that was farmed in New Zealand. There's also the fact that it is a uniquely New Zealand species, which means we wouldn't be competing with Asian commodity markets that have already been established. And we're also looking at the fact that it's on commercial fishing licenses already, which means it's easy to implement regarding the current legislative environment here in New Zealand. So yeah, those are a few of the reasons.
I mean, you talk about, the balanced ecosystem, but isn't harvesting or growing going to have a negative effect somehow? All aquaculture does have some kind of effect on the natural balance, does it not?
Yeah. So I think what's actually important here is that because seaweed doesn't require any inputs itself, so you don't need to apply fertilizers to it for it to grow, it actually takes up nutrients that are in the water that have been excreted from, say, other farming processes. So mussel farms or salmon farms, they've got waste from their species, and those nutrients can be absorbed by the seaweed. And it's also important to note that seaweed is a natural habitat for many species, and research overseas has already indicated that in and around seaweed farms, you get increased bioactivity and biodiversity. And it is unclear whether how that affects mussel farms, so the increased biodiversity around the mussel farms, but that is the course of some current research here in New Zealand.
I mean, if you got all your seaweed sucking up the waste from your mussel farms and your salmon fishery,
yeah,
do I want to use that seaweed? Is that not going to affect the nutrient content of it, for example?
Yeah, so seaweed is going to absorb whatever's in the natural environment anyway, and it's very hard to track, what is in the environment and what it has absorbed. But the compounds and the nutrients and the inorganic and organic components of seaweed have been shown to be very useful in a range of applications. And once you've got the seaweed, it's very much about how you process it and how you extract compounds that you're after. So you're not necessarily using the whole plant itself for certain purposes. But yeah, I think it's important to note that it will suck up anything in the environment that it grows in regardless. So it'll be hard to quantify that where it's come from anyway.
So you want to grow it effectively on the existing mussel farm?
Yeah, so the model we were looking at, so it's not using the mussel lines that are already there, but it's integrating it with existing mussel farms. So you'd have similar technology in the fact that you've got long lines that are set out, say, 10 meters apart from each other, and then the seaweed would be submerged underwater and it would grow up, and that would be, say, next to the mussel farming. It's not like there's a row of mussels and a row of seaweed. I'd imagine it would be seaweed over here and then mussels over here, and depending on how much space you have, they could be a significant distance apart in such a case that if you're worried about some of the effects of absorbing the nutrients from the mussels, you wouldn't have to worry about that specifically, but you could still use the same resources in terms of the boating infrastructure, and those kind of things.
I'd be interested to know whether there was any difference between the benefits, the content of the wild seaweed versus the farmed seaweed, and as you say, depending on where it was farmed.
Yeah, I do think that is again a topic of current research, but I think importantly, the species that you farm are a lot more reliable and consistent with the nutrient content that they have, because they're a specific strain. And when you actually go to put out the the seaweed lines, you've actually seeded them yourself, so you know exactly what strain is on there, and you get a lot more transparency and consistency with supply as opposed to wild harvest. So I'm not too sure whether there is a difference between the nutrient content, but I know it's a lot more consistent with farmed seaweed.
In your presentation, you talk about pesky red tape inhibiting the development of the industry at the moment.
Yeah.
Care to be specific?
Yeah. So pesky red tape might have been the wrong choice of words, but it's more that there's not specific reference pertaining to seaweed aquaculture in like the Fisheries Act, for example. Like we don't have current legislation guiding how a hatchery would work, whereas we do have guidance on how a hatchery would work for mussels and for salmon and those species. And what we're really looking for is researchers to become more aware and understand what we need in the legislation, and that can guide MPI forward with producing robust legislation to develop the industry, because MPI introduced seaweed into New Zealand in terms of aquaculture. So yeah, it's just figuring out what we actually need to have in the legislation for it to be a viable and profitable industry.
And the first thing would be to get the buy-in from the mussel farmers, would it?
The buy-in. So yeah, it's not necessarily not necessarily a buy-in situation, because like the model that I've suggested is just one of, say, 1,000 different models that we could have for introducing seaweed aquaculture into New Zealand. In fact, recently, actually, I think it was a couple of days ago, a Kunley radiata has been approved to be the first farmed species commercially in New Zealand, and that's going to be taking place in the Hauraki Gulf, and that is with existing mussel farms using their technology to test that out. So yeah, we're getting an industry off the ground, and I think the relationship with mussel farms is very important, but that's only one model again. So yeah.
Excellent. Thank you very much.
Right. Thank you.
Thank you, Robbie. Our next finalist is Sharland Shah. Sharland is studying engineering science and global studies at the University of Auckland. He's always been curious about science and technology. That, combined with his interest in public speaking, has created his interest in science communication. Outside of his studies, he has a passion for acquiring more knowledge about literally anything and enjoys exploring Aotearoa's beauty and nature by hiking. Sharland's STEM presentation is MR1-based cancer targeting, a one-size-fits-all approach. Thanks, Sharland.
Thank you, Joy.
Kia ora, Sharland.
Kia ora.
If your T cell targets MR1 protein, and MR1 protein is on all cells, how do you limit it to attacking only the cancer?
That is exactly what we need to find out. So that's one of the biggest, so far experimental evidence suggests that the T cell doesn't do that. The T cell was remarkably good at leaving healthy cells alone. I mean, in fact, not only healthy, the researchers infected some of the cells with salmonella, and it still didn't touch them, and it only got activated by cancer. So it's sort of a black box at the moment, and that is one key issue or hurdle that we need to clear before we get ahead on this specific receptor target when it comes to T cell therapies.
Because otherwise it's going to be no improvement on chemotherapy or radiotherapy, is it?
Definitely. I mean, T cell therapies can still be an improvement as a whole, but this specific receptor, if you can't target that, or if you need to understand how and what it is that it's sampling from the MR1 receptor and how it communicates with it specifically. And once we understand that, we'll have more insights. And there's reason for optimism there, because there's only been one study. This breakthrough was discovered in January this year. So things are still picking up for this.
The Malligan Institute, as you point out, has been trialing CAR T cell therapy, but not targeting this particular protein. Is that different?
They're using a mechanism called TLR2, and that is mostly, so TLR2 can be used to identify quite a bit of things, but there's some research suggesting that in cancer, it's it's sort of turned off in a way, and that's how cancer avoids it as well, avoids the normal T cells looking for it. So we can train the T cells to find the avoidance of or the inhibition of TLR2 and then identify cancer.
I mean, interestingly, this would be not only a treatment, but also prevention, would it not?
Yes. So it would. This is definitely in line for bringing in cancer vaccines. The only thing with it is that cancer isn't a set thing, and so we can't design a specific vaccine for each and every cancer we find. It's just going to be way too big. We need to find that receptor that will work for multiple, if not all, cancers, and MR1 has promised that to us so far.
Why do you think what was the process of this discovery? I mean, did they try and target a whole range of proteins and they found one that worked?
Funnily enough, they weren't looking for cancer targeting cells. They were actually trying to see how to destroy bacteria with T cells, because T cells are killer cells. They can kill anything. And the reason why they were growing the bacteria on cancer cells was because cancer cells are resistant and they're hard to kill. So they'd know if it was working on the bacteria or not. And surprisingly, this one killer cell that they named MC7G5 ended up killing the cancer, and I'm assuming not the bacteria, because later on it didn't kill infected healthy cells. So that's how they stumbled upon it. And then they used CRISPR to identify what exactly was the receptor. So they started inhibiting certain receptors to see what it is that it's interacting with, and that led them to MR1.
The backstories are often more interesting than the outcome.
Definitely.
This is genetic modification of a human being, though, essentially, isn't it?
It's genetic modification. Yes, it's a genetic modification for the cells, for the cells that are either donated or created in a lab. So there's no shying away from genetic modification here. You need that. That is core to T cell therapy, because you need to program the receptors in.
Is that an obstacle?
That is an obstacle for New Zealand, yes, because I think we've got this really inherited view of against genetic modification. And in all honesty, this might be something because I'm a young person, but I haven't seen a debate for it. It's just been this inherited negativity against it. And the truth is that within the past 25 years, so the HSNO Act was signed in 1996. Since then, we've had quite a bit of progress in genetic modification and gene editing. And I think we ought to update our stance and change the status quo by having a new discussion of looking at the facts now as they stand and what the benefits are for us.
How expensive would it be for us to start seriously investigating this, rather than leaving it up to, better funded places?
So CAR T therapy is expensive right now. Currently, so the trials that Malaghan Institute is running, they're going to take up to 12 patients, and they're estimated the cost to be around about $14.4 million, which is quite a bit of money for 12 patients. And when it comes to research, it's a bit hard to put a number on it, because it depends on how far we need to go and how much we need to do, but it is capital intensive from looking at previous records. But looking at the benefit that it provides, it's nowhere, it makes sense to be making that investment. So an independent economic research organization called NZIER estimated that within the next 20 years, if we just use T cell therapy for lymphoma, so non-Hodgkin's B-cell lymphoma, and to treat that, you'd be saving around about 2,812 lives, which would be worth about $7.5 billion for New Zealand. So in the long term, spending on research will pay off, and evidence of this can be seen in UK, where they did a cost analysis where every pound spent on cancer research has historically returned about 40 pence a year. And to give it in context, governments generally find it a good investment if a dollar spent returns about 3 to 5 cents a year. So 40 cents a year is 40 pence. That was in the UK, but I think that we can draw similar parallels between the UK and New Zealand. Yeah.
The resistance is a problem with CAR T cell therapy because you're targeting a single antigen, right?
Mm-hmm. So there is the fact, there is the possibility of mutations, but with MR1, what's so good is that it's related to so many other processes in our body, and it's found everywhere that it makes it hard for a mutation to be carried out into someone. Like within someone, there might be a thing, but as a general case, that's less likely than a mutation into a small thing, because a mutation in MR1 will affect a lot more things than just this cancer targeting cell.
Which brings us back to the original point that you
So,
don't want every cell affected by the CAR T cell.
No, so definitely we need to understand what exactly it is, because MR1 is sort of this sampling site. So it essentially is like a hole into the cell where, well, not a literal hole, just where it can look through and see what's inside the cell. So we need to figure out that what is activating this T cell and make sure that we understand it more. And that is absolutely crucial. Most of the weight relies on that, but there is optimism since it's not like we've exhausted our efforts. We've only just started and there's a long way to go into it. And I think New Zealand is in a good position to actually pick up on it.
Thank you.
Thanks a lot.
Thanks, Sharland. Our next finalist is Sophie Mance. Sophie's a first-year physics student who started the year at the University of Otago before working at Victoria University's Wellington Robinson Research Institute until her move to England 2 weeks ago. She's currently in quarantine before she commences her studies in physical natural sciences at the University of Cambridge on a 2020 Girdler's Scholarship. She's represented New Zealand at the 2019 International Young Physicists Tournament and is also a member of the Under-20 Women's Ultimate Frisbee team, Kahu. Sophie is passionate about the environment, so when she's not in a lab working on a project or out in the field, she'll be in Frisbee or she'll be volunteering with groups like Climate Change, organising climate change conferences for high school students or Generation Zero. Sophie's STEM presentation is Aspergopsis amate to reduce methane emissions. Thank you, Sophie. Thank you, Kim.
Kia ora, Sophie.
Kia ora.
How much of this one species of seaweed would we need to produce, do you think?
A lot. So per cow per day, you're looking at about 100 grams. And so we've got
100
about
grams of seaweed or 100 grams of the extract?
I believe it would be 100 grams of the extract, so more of the seaweed. So given that we've got about 10 million cows in New Zealand, and I just did some quick back of the envelope calculations on this. So in my presentation, I came across the figure 67%. If 67% of New Zealand cows essentially were using this additive, then that would fulfill our zero carbon act commitment. So that gives us a figure of 677 tons of dried extract a day. So we are talking enormous amounts, and so it's likely that we're going to need to, it's going to take time to be able to reach that level of production. But yeah, so we're talking significant quantities, but we are also talking about an enormously effective product.
Yeah, but I'm just wondering how much of our ocean, our coastline, wherever, we would need to devote to it.
I'm afraid I don't know what the land, well, not the land, what the space requirement would be. But the other thing to note is that whilst it's a native species to New Zealand, It's also found in other places around the world. So it's not like New Zealand would need to be the only place in the world producing this. So that means that there is no pressure, I think, to intensify beyond what the what can naturally be sustained in the area, and no need to go beyond, no need to harm our natural ecosystems.
One of the pluses for it was, you say, its export potential, though.
Yes. Yes, well, currently the way I see it, and when I was fortunate enough to talk to some of the founders of the company CH4 Global, is that it's got export potential. In New Zealand, we're in a good position to be able to start producing this quickly. Their CH4 Global expect to have a product by the end of 2021. So if we can start as one of the producers of this and exporting that to countries where there's a real demand for it and where we can sell it for a high potential, that's going to have benefits to New Zealand. And then over time, as demand increases, then that's when you look to perhaps trying to work with other countries and help them develop their own systems whilst maintaining a sustainable level of production yourself.
So CH4 Global is doing its operations on Rakiura, despite the fact that the long-term effects are unknown of this.
What I meant by the long-term effects unknown being the long-term effects in the cattle are unknown, and what I mean by that is it's possible that if you continue to feed the cows the additive over time, the methanogens may be able to adapt to it. And so that's what needs to be investigated further. But initially, there doesn't seem to be any evidence of this at inclusion rates over 0.2%.
Yeah, the maximum is 90 days trial so far. Not a lot, right?
No, not a lot. But given that, so what they noticed was that at a 0.05% inclusion rate, it did seem to become less efficient. But at the 0.2%, this is in just one of the studies, and the 0.2% inclusion rate, then they didn't notice this effect. And considering what would have to occur, there would have to be another pathway for methane production. Considering that all of these species of archaea use the same one, it seems unlikely to me that that would develop. And so I think that it's safe to assume for the moment that this is going to be an effective solution. That's my
opinion. Can't we just make bromoform? Why do we need to grow seaweed?
So the reason for that is use of artificially formulated HMAs is banned because of their ozone depleting effects. My understanding of this is that it comes in the production of these is harmful to the environment, and also when the bromoform's in the seaweed, it's at a low concentration and it's also contained within the seaweed, and so it's not going to be getting into the atmosphere.
If you give it to cattle, are you sure that they'll remain healthy? It sounds like quite a potent toxin.
So we're talking about very low percentages. There have been the two potential causes for concern would be if it's getting into the meat or the milk, and studies show that it's not. And the other concern that I highlighted in my presentation was the shift in the acetate to propionate ratio. But that can be caused by other factors in the cow's diet as well. It's not a unique symptom just of this additive. And it's just something that farmers need to be aware of so that it can be managed. And many of them, it's something that they'd be aware of and monitoring anyway within their cattle, just like other changes in feeds could lead to this change in volatile fatty acids ratios. It's just something to be aware of.
Is there, okay, inhibiting methane production, good, but is there anything else that's affected if methane isn't produced?
So as I said in my presentation, we're going to be increasing, there's going to be an increase in the carbon dioxide emissions of the cattle. So it's not a complete solution to biogenic greenhouse gas production. So we are going to need to look to transition away from agriculture, but considering that methane is far more potent than carbon dioxide, and if we want to get climate change under control, we need to act rapidly and drastically. it seems to me like this could be a good solution and a good way to bring our methane emissions down.
But the main application would be to extract the bromoform from the seaweed and then give it as a supplement?
No, so the seaweed is just freeze-dried, so grown and then freeze-dried directly. So that means that there's not a huge amount of processing costs,
Right.
I think, which makes it quite appealing. Yeah, so it's quite a simple process.
Do they like it, the cows?
They don't seem not to like it, and actually it's cows eating seaweed is actually not as bizarre as you might think. It's been going on for a long time, so that doesn't seem to be a barrier.
Nice to talk to you. Thank you, Sophie.
Thank you.
Thank you, Sophie. Our last, but by no means least, undergraduate finalist is Isla Day. Isla is in her second year studying biomedical science and physical geography at Victoria University. Her interest in science is strongly influenced by her love of the outdoors and motivation to protect the environment. Isla was the key organiser for the School Strike for Climate movement in New Zealand and now coordinates the Wellington branch of the Vote Climate 2020 campaign. She is also a keen mountain biker, riding for fun as well as racing and instructing. Isla's STEM presentation is Hydrogen by Electrolysis for a Fossil-Free Future. Thank you, Isla. Thank you, Kim.
Kia ora, Isla.
Kia ora.
As you point out, only 5% of our emissions, our greenhouse gas emissions in 2018, came from energy generation. So why not focus on another area? This seems to be largely solved.
Yes, New Zealand does already generate a very large proportion of our electricity from renewable sources. What's so appealing about hydrogen is not only that it helps us eliminate those emissions from electricity generation by solving the dry year problem, but also by creating those co-benefits and helping us decarbonize the rest of our energy sector. So the Interim Committee for Climate Change had identified electrification as the key solution to decarbonizing the rest of our energy sector, but it can't do it alone, as there are some cases where electrification can't be used. For example, in long haul transport, so like planes and ships, they can't stop to recharge mid-journey, whereas hydrogen could be used in this capacity. Also, New Zealand has a lot of rural areas or areas that may not be connected to the electricity grid, where hydrogen could also create a more equitable transition away from fossil fuels in the energy sector. It also provides numerous social and environmental benefits. And so it's not really just about eliminating emissions from the electricity sector, but also the co-benefits that come along with using hydrogen technology. It's really just the tip of the iceberg, eliminating those emissions.
How would it protect against the dry years? You talk about the dry year problem.
Yes, so the dry year problem states that obviously renewable electricity is dependent on environmental conditions that vary from year to year. So New Zealand's just experienced an abnormally dry winter, as we've seen, and the Auckland dams have been particularly low. So if we had energy stored in the form of hydrogen, we wouldn't need to worry about the fact that we can't generate as much electricity from hydropowered dams. Or if it's not windy, we wouldn't have to worry about not being able to generate as much electricity from wind farms. So this energy can be stored as hydrogen during wet years where we may have more rainfall or more wind, and can be stored for these dry years. So we don't have to fire up natural gas peaking plants, which is what currently fills this gap, is burning natural gas. So to avoid those emissions, we can store the energy as hydrogen and then use that in years where we can't generate as much renewable electricity.
How can it be stored?
There are a couple of ways that you can store hydrogen. Some of the most common ones are storing it in high pressure tanks, so as a liquid. But a really common way to store hydrogen is by converting it to ammonia, and then it can be reconverted to hydrogen for use in hydrogen fuel cells.
Danger is one of the things that people worry about with hydrogen storage, is it not?
Yeah, so hydrogen in itself is quite flammable, slightly explosive. The Heisenberg Zeppelin's a good example of this. And so it'll really be a case of how well we can store this. And that's one of the reasons that it's often converted to ammonia, because ammonia is less hazardous. but there are energy losses associated with this conversion. So there is research at the moment looking into how we can make these storage systems more efficient at limiting these energy losses, so we can store it as ammonia and don't have to worry about hydrogen being so explosive. Yes.
Why don't we just concentrate on making better batteries?
So batteries don't solve the problem of electrification being quite inequitable. So if we make better batteries, to make these batteries, we're going to have to extract lots of rare elements that are, and their extraction is really unsustainable. They're coming from countries that don't have the proper safety regulations, which is creating some human rights issues, as child and forced labor is having to extract these compounds. So using a solution in New Zealand, created by generating hydrogen rather than relying on better battery technology, will help us solve our own problems rather than exporting our problems to other countries and creating wild social inequalities. Yeah.
You imagine exporting the hydrogen there?
Yes, New Zealand's super lucky that we have great opportunity to generate renewable electricity. We live in a mid-latitude, so we get frontal weather systems, so lots of wind and rain, and also New Zealand has a lot of space where we can do things like build wind farms or like create hydropower dams. So New Zealand's really lucky that we can tap into this infinite resource that is renewable electricity, whereas other places may not have the same ability or may not have time. A lot of countries are running out of time leading up to their commitments in the Paris Accord to meet their emissions reductions targets, and they don't have time to build these dams or create hydrogen to decarbonize their energy sector. So New Zealand could capitalize on this and become a global leader in generating green hydrogen and exporting it to other countries that don't have the same opportunities as New Zealand does.
You talk about Mātauranga Māori. What is the aspect of this that addresses that?
I think it really hinges on climate justice and ensuring that the communities that are going to be hardest hit by climate change, in particular Indigenous communities, aren't being harder hit by an inequitable transition. So by creating a just transition, we are empowering these Indigenous communities. And that starts with creating partnerships and integrating Mātauranga Māori and the Māori worldview of sustainability into our solutions. We have a great opportunity to create partnerships with iwi groups who are the rightful owners of the land that we are considering constructing these renewable electricity generators on, to create economic benefits for them as well, and collaborating with these groups and taking knowledge from Te Ao Māori and Mātauranga Māori is a great start for this.
The cost of this, you would need, as you pointed out, to repurpose the infrastructure, wouldn't you? Which is massive expense.
Yeah, absolutely. There are definitely costs associated with integrating hydrogen technology. But unfortunately, in solving the climate crisis, not everything is going to be free. And hydrogen is approaching economic viability and will keep on doing so as demand for export increases and our storage solutions become better. I think it's also important to consider the long-term economic costs of us not averting the climate crisis, and also the current environmental and social costs that we're even currently experiencing. Environmental costs like more frequent droughts, such as we've seen in Auckland, or unprecedented wildfires as we're seeing in North America, and the social cost of our Pacific Island neighbours facing a future where their islands are possibly underwater. The 27th of September global protest last year, the largest protest per capita for school strike was actually in Tokelau, where 20% of their population turned up. So they're facing these social costs already. And New Zealand has an obligation to take action, and we should not be the ones to tell the Pacific Islands, like those in, like the people in Tokelau, that we chose not to take action on climate change because it wasn't profitable enough for us.
Thank you.
Thank you.
Well, that's the last finalist in this afternoon's question and answer session. Have any of the judges any additional comments they'd like to make? Over to you, Margaret.
I don't think at this particular stage, Joy, I think we want to have a chance to discuss together, and we'll get some generic feedback for the participants when we do the announcements in October.
Thanks very much.
But I did just want to add, sorry, that I did appreciate their adaptability, both in terms of changing to the online format, which I thought people did really well, and into their answering the questions in a way where they're decoupled from the presentation. So, you know, congratulations to all of the participants.
Thanks, Margaret. And thanks again to the judges, to all of you for your time, you know, your expertise, your dedication, and your support from Eureka. We really do rely on you, and thank you so much. And thank you also for the finalists, as Margaret has said, for embracing that change to the online format. I think certainly the questioning sessions and the depth of the questions and the students' far more full answers, I think has been because it's separated from the presentation where it's normally immediately after it. So it's been very interesting to sit and listen to this year. Thanks again to our sponsors, because once again, the event would not be a success without your support. And I think the events of the past year highlight now the importance of robust science, research, and the strong, understandable communication when it's delivering a complex topic to a general audience. We certainly need more communicative from the science community than less. So again, we thank Science New Zealand, the Ministry of Education, MB, DOC, MPI, NIWA, Ministry of Health, High Value Nutrition National Science Challenge, Federated Farmers and South Pacific SERA, MSD, Becca, Zone Law, Chris Parkin, and the McDermott Institute. And finally, thank you to all of you that have been watching. We appreciate you joining for this session, and we look forward to sharing the results and celebrating with these amazing young STEM students and following their careers as they excel in their chosen fields. So the webinar will now end. Thank you all.